Hello viewers, you are watching me. I'm Zahur Islam. Today I will teach you about enzymes and its basic concepts. So look at here, this is the content, and this content we will discuss about the basic chemistry of enzyme, classification of enzyme, mechanism of enzyme actions, enzyme kinetics, inhibition, activation and specificity. So now come toward the basic introduction of enzymes. Enzymes are biological catalysts that speed up the rate of biochemical reactions. As we know that most enzymes are three-dimensional globular protein, tertiary and quaternary structure. Some special RNA species also act as enzymes and are called ribozymes. For example, hammerhead ribozyme. So you can see here. So look at the structure. So this is hammer-like hammerhead enzymes. So this is very easy to understand. So enzymes, they are capable to enhance the biologicals, the biochemical reactions that take place inside or outside the body. So naturally this is biological catalyst, how it can speed up the chemical reactions. Come toward the structure of enzymes. If we can review the overall structure of enzymes, there are the active sites of enzyme is the region that binds substrate, cofactors and prosthetic groups and contain residue that help to hold the substrate. The active sites generally occupy less than 5% of the total surface area of enzymes. So as we know better that there are some particular active sites, and with these enzymes can act, and these enzymes can act and bind with the substrate, cofactor, prosthetic groups and contain residues that help to hold the substrates. The active sites has specific shape due to tertiary structure of proteins, while a change in the shape of protein affect the shape of active site and function of enzyme. So this is very clear. Look at here. This is the active sites. So active sites can be further divided into two. One is binding sites, the second one is catalytic sites. Binding sites, it chooses the substrate and bind it to activate the sites, while the catalytic site, it performs the catalytic action of enzyme, that how the enzymes perform their catalytic activity and how it can speed up the chemical reaction. So there are some particulars active sites, and with these enzymes acts which are called substrate. Now what are cofactors? So cofactors can be defined as these are the non-protein molecules which carry out the chemical reaction that cannot be performed by standard amino acids. Cofactor are two types. One is organic cofactors, while the second one is inorganic cofactors. Now let's see here in this diagram. So you can see this is cofactor, while this is inactive proteins. Without the cofactor attached, the protein is not active. Whenever there is attachment of cofactor, which is the non-proteinaceous part, so it is also very essential, very important to carry certain type of chemical reactions which are not performed by the standard 20 amino acids, and then inshallah I will discuss it in detail. So it means that without the cofactor attachment, these proteins, they are not active, while in the second case, in this case, the active proteins, these are the binding sites, this is L1 and L2. So these are actually, these are the cofactors and these are the binding sites. So they are actually bind here and it can so activate this protein. So cofactor binding activates this protein. So if we can compare these two sketch with one another, so here the cofactor is without attachment. That's why without attachment, without cofactor attachment, the protein is not active. While in this case, the cofactor that is binds, these are the binding sites. So when it is binds with, you can say, this cofactor with enzyme, so it can activate these proteins, which is the non-proteinaceous parts. As I have already told you, that it carry out certain type of chemical reactions. While the inorganic cofactor, these are the inorganic molecules required for the proper activity of enzymes. For examples, enzyme carbonic anhydrase, this is the name of the enzymes which requires zinc for certain activities like hexokinase, his cofactor, which is magnesium ions. Organic cofactor, these are the organic molecules required for the proper activity of enzymes. Let's suppose, for example, the organic cofactor, so the classical example is glycogen phosphorylase, which required the small organic molecules, pyridoxal phosphate. Okay, now let's see here the types of organic cofactors. So it is being classified into two categories. One is prosthetic group and the second one is coenzymes. So prosthetic group is actually a coenzymes is loosely tied bound organic co-bounds, organic cofactors, and factor, for example, that is flavins, heme, and nicotinamide adenine dinucleotide groups and biotin like vitamins. So the non-proteinaceous part is also called prosthetic group, which is also very important upon the activity of enzymes. So this the coenzyme, it is loosely tightly bound to that organics cofactors, especially the cofactor or factor like flavins, heme. So these are very important. So look at here. So this is a helical shape of polypeptide molecules, and this molecule you will see the iron, the alpha chain, and this is heme group, or you can say beta chain is also present. So this is, you can say, the globular structure, or you can say the helical shape of the polypeptide molecule, while this one is nicotinamide adenine dinucleotide NADPH. So this is the the geometry, the structure of the nicotinamide adenine dinucleotide. So it contains, you can see this is a nicotinamide, while this is ribose, and diphosphate bond is present here, while this is also ribose and adenine and plus charges. So it means that as we can say at the same case like a chemical bonding, so when they are attached with one another, so the same situation is here. And enzymes with a cofactor remove as kinetic as apoenzyme. So it can be represented as apoenzyme, while the complete complex of a protein with all necessary small organic molecules, metal ions, and other components is termed as holoenzymes or holoprotein. So you can see here, this is another structure of holoenzymes, and this is the proteins chain. You can say this is the protein chain. Protein chain can be observed here. So these are the active sites in which usually these enzymes can directly act on the substrate. And this is cofactor, which we have already discussed. Now come to our substrate. So inshallah in my next lecture, so I will discuss about the substrates, that how substrates they are joined with the other molecules, how the enzymes act on the substrate. So we will discuss all these things in my next lecture. Thank you so much for watching my lecture.